All right. Hello, everyone. Can you guys hear me? Yes. Awesome. So today we're talking about molecular evolution and phylogenetics. And I love this slide that Patrick Winston likes to use, where somewhere something went wrong. So we are basically in the second half of the comparative genomics and evolution module, talking about molecular evolution, tree building, and phylogenetics. And then next time, we're going to be talking about phylogenomics. So today we're going to be talking about evolutionary rates and models of evolution, and also how we build distance-based trees, how we do dynamic programming on a tree, and Bayesian models for phylogenetics. So as you know, the tree of life is extremely complex. And just one second, I'll share. And it all boils down to some initial life form that all modern life that is visible today has evolved from. And this is a pretty remarkable feat. It's not like life arose a bunch of times. No, life arose once, and then that diversity can be traced down all the way back. And these trees are basically the way that we can understand this hierarchical relationship between, plants and animals and, arthropods and fish and reptiles and mammals and, human all the way down there. And you can see these morphological similarities and the different evolutionary innovations that are, demarcating each of these major lineages of life. But life is also full of extinction. So, here we are today. And the only reason why, mammals are around today is because 65 million years ago, this giant asteroid basically hit the Gulf of Mexico in Chicxulub and resulted in dramatic impact of both short-term effects of just wiping out big species, but also long-term effects of climate change, possibly leading to all of these giant animals no longer being able to sustain themselves that way. And if you look at, human evolution, we have been ruling the planet for a tiny little amount compared to dinosaurs that were ruling the planet for, 130 million years before we showed up. So these species have now expanded into, of course, birds and crocodiles, which are just a tiny little lineage of this incredible diversity of dinosaurs. And what we have in phylogenetics is the challenge of, well, we only have modern species to work with, or in some cases, we have bones of extinct species. And then the general problem that we're trying to solve is how do we infer a complete ancestry of a set of quote unquote objects based on knowledge of their quote unquote traits. Now, these objects can be species, genes, cell types, diseases, cancer types, languages, faiths, cars, architectural styles. They could be anything. And the traits can be morphological traits or molecular traits or gene expression or TF binding or motifs or words and, types of records that someone listens to and so on and so forth. And then the historical record varies dramatically between fossils for extinct species and then imprints for even further extinct species that didn't have hard bones. The timing of different geological events, they can basically tell us that this entire lineage was formed after, the South American continent merged with the North American continent, for example. And also, in some cases, living fossils. Basically, these are species that have changed extremely little since their ancestral forms. But also sequencing of, in some cases, extinct species based on whatever DNA remains over, a few thousand years. And of course, paintings and stories and so on and so forth. So today's lecture is going to be focusing on phylogenetics using only extant species data. So these are species that are still alive today. And then focusing on gene trees through paralogs, orthologs, and homologs without distinguishing the three. Whereas next lecture, we're going to be talking about, specifically gene trees and species trees jointly. And therefore, we're going to actually be distinguishing between paralogs, orthologs, and homologs. So the plan for today is, number one, introduce the basics of phylogenetics, basically, define it and introduce it, what are characters, traits, nodes, branches, etc. And then look at how we build alignments, how we build distance matrices from alignments by modeling sequence evolution. How do we build trees from distances through tree building algorithms? How do we build trees from alignments directly by scoring the alignment that we observe given a particular tree? And then how do we explore tree space through this Markov chain Monte Carlo search? So let's dive right in and look at what are these basics of phylogeny. So we're going to be talking about characters, traits, gene, and species trees. So trees can be inferred by many criteria. The traditional criteria have been morphological traits. So basically phylogeny existed well before molecular data was available. And the type of data that was being used is, for example, do you have a particular breathing hole in this part of your skull? How many teeth do you have? What is the relative shape? And so on and so forth. Now, these traditional traits were usually very sparse. There were very few traits, and they were usually very well behaved with very little back mutations. Whereas modern traits are based on enormously more data. So basically we can have, one nucleotide for every position in the genome is a trait, but every one of these traits is actually less reliable. So basically traditional phylogenetics was all about building species trees only. And it had a small number of traits like hoofs and nails and teeth and horns. And these were well behaved with the easy assumption to make that was true most of the time, where each of these traits only arose once. And this principle of parsimony was very usually used that assumes Occam's razor, namely that I will assume the fewest number of events in my phylogeny. And if there are more events, I will reject it for an alternative explanation that has fewer events. So Occam's razor is basically the idea that if I have two hypotheses that explain the world and the data equally well, I'm going to choose that hypothesis that is less complex. And that's, applies in the case of parsimony for phylogenetics by basically saying I'm going to choose the topology that has the fewest events. So that's traditional phylogenies. They're only able to build species trees, and they're based on morphological traits, which are few but well behaved. Modern phylogenetics is building both gene trees within a species and across species, as well as species trees when you have one gene for the same gene across each of the species. It uses a very large number of traits where every DNA base and every protein residue could actually be a quote unquote trait, namely what is the residue at that position. And these traits are frequently ill behaved. Back mutations are enormously frequent, and that can be perceived as convergent evolution, whereas with these traditional traits, convergent evolution was much more rare, namely the fact that two different species evolve the same trait, even though neither of them had it before, independently is, very rare. And the reason why we call it convergent evolution is that most evolution is in fact divergent. You start with a common ancestor, and then most of the changes bring you away from each other. But convergent evolution, for example, if this lineage here and that lineage here both evolve, wings at the same time. So, bats and birds might evolve wings and flying at the same time. And this basically could be considered as bringing the traits closer to each other, hence convergent rather than divergent. So who's been so far on this part of the lecture? Great. So we're at 100%, actually 90, 10, 0, 0, 0. So again, these back mutations are frequent, and they are not just simply convergent evolution. They look like convergent evolution, but they're simply, coalescence events or convergence events. And there's also a very small number of letters that arise many times independently. And therefore, it's much more common that, for example, a T changes into an A, then changes into a G, and then they both change into a C. So that's an example of conversion evolution, but it's very, very common that this happens because there are only 4 letters. There's only 4 states that you could be in, and therefore over evolutionary time, you're very likely to have these conversion evolution. So let's go over some definitions. So we're going to be talking about the ancestral node or the root of the tree. We're going to be talking about internal nodes or divergence points, and these are representing hypothetical ancestors of the taxa. We're going to be talking about branches or lineages for each of those, and we're going to be talking about the terminal nodes or leaves for the leaf nodes that are observable today. So these represent the different taxa, genes, population, species that are used to infer the phylogeny. So most of the time, we're going to be having information only here. We're going to be inferring probabilistically the information in the ancestral nodes. We're going to be distinguishing 3 different types of trees. The cladogram is only worrying about the topology, where the specific branching points and lengths of the branches don't matter. It's just about topology. Chronogram is assuming that every species has evolved the same amount of time since the common ancestor, and is only worrying about divergence times between them, so chronological events that cut across that tree. Whereas a phylogram is including the topology as a cladogram, the divergence times as a chronogram, but also the different divergence rates of each species. So for example, taxon B might be evolving 6 times more rapidly than taxon C, even though they've both been on the planet the exact same amount of time, just like everything else on our planet. They've been evolving at a faster rate at the molecular level, and we can represent that in a phylogram. So our challenge today is going to be, how do we infer a tree from a set of nucleotides and peptides that we have aligned using any of the methodologies that we talked about so far? So the first question is going to be, how do we use sequencing data, such as nucleotide alignments and peptide alignments, to infer the evolutionary history that represents all of the bifurcation events that gave rise to each of these lineages? Again, this could be species, this could be genes, or any other kind of thing. So for example, this could be a single gene family within the human genome, or it could be a single gene across a bunch of related species, or it could be a combination of both multiple genes in the same family, but across multiple species. So that's going to be the goal for today. So how do we build these molecular phylogenetics methods for inferring the evolutionary history from a set of sequence data? And the way that we're going to be doing that is in 2 steps. So basically, either we're going to go from the sequence alignment to a pairwise distance matrix and then build a tree from that matrix, or we're going to be looking at these character-based methods that are going from alignment directly to phylogenies. But these methods are mostly a scoring set of methodologies. So we need to couple them with tree proposal and scoring to basically find the topology. So these methods will be able to score the topology really well, but then we're going to have to search over topologies in order for each of them to be scored. Whereas these methods here basically are summarizing this enormous amount of information in the alignment into just one number between this entire sequence and that entire sequence. And that one number at every pairwise comparison is then used to infer the tree through these tree building algorithms. And we're going to talk about multiple different types of tree building algorithms. So who's with me here on the outline and the goal? Okay, so we're at 70, 30, 0, 0, 0. All right, so first we're going to be looking at how do we go from sequences to distances, then how do we go from distances to trees, and then how do we go from sequences to trees, and then lastly, how do we propose and score these trees. And these are going to be the four components of the lecture. So we talked about the basics of phylogeny, basic introduction and definitions, and now let's turn to how do we go from alignments to distances. How do we model sequence evolution? How do we turn pairwise sequence alignment into pairwise distances? And how do we build probabilistic models of divergence? And in particular, the Hughes-Cantor model, the Kimura two-parameter model, and then a hierarchy of models. So this is the first part of going from distances to, from sequences, alignments to distance matrices. All right, so the question is, how do we measure the evolutionary rate if I have an alignment of two sequences over, say, 1,000 nucleotides? How do I measure the distance that this represents? I could simply compute the overall nucleotide divergence. I could assume a uniform rate of divergence and simply report what is the overall percent identity. Okay, so, if it's 90% identical, I can just say, okay, the distance is 10. And if it's 20% identical, I can say the distance is 80, and so on and so forth. So that's the simplest one, just looking at the overall percent identity. If I want to be a little more fancy, I could say, wait a minute, transitions are much more common than transversions. So A to G changes very frequently, and C to T changes very frequently, because these are both purines, these are both pyrimidines. They actually can change into each other much more easily through polymerase making mistakes, because these bases are bigger and these bases are smaller. So it's actually easy to confuse them. You can sort of swap one for the other. So that's modeled through a two-parameter model that we're going to see that actually allows for transitions and transversions to have different rates of interchange. Another model could be even more fancy to basically look at synonymous versus non-synonymous substitutions. So we could basically ask, given our genetic code, and given that any of these four any of these four codons can all encode the same amino acid, then I can evaluate how many amino acids are, how many nucleotide substitutions are amino acid changing versus how many substitutions are amino acid preserving or synonymous. And we can ask, what is the ratio of amino acid change substitutions over the total number of substitutions, or the total rate of substitutions inferred by the synonymous sites, to basically specifically look for positive selection or for functional selection, and so on and so forth. Okay. Who's with me so far on the different ways of measuring divergence? So we can measure either raw nucleotide divergence, or distinguish transitions and transversions, or actually distinguish synonymous versus non-synonymous changes. So 100, 0, 0, 0, 0. Okay, so the next challenge is that the number of actual mutations that occurred is always greater than the number of observed substitutions that we see in the alignment. So if I see an A aligned with an A, for example, so if in my alignment I see an A here and an A there, and a T here and a T there, for example, then the Occam's razor approach would basically say, oh, great, this A has been conserved, this T has been conserved, and so on and so forth. But what's actually happening is that the A over large evolutionary distances was probably at some point changed into another character and then changed back into an A. Let me explain. If you look at even just one evolutionary time, if you look at only one evolutionary time step, and I start with an A, I can basically change into a C, a G, or a T with some probability. But then it gets interesting at the second time point. I've now taken this 0.1 probability jump and I'm at the C. The question is, where do I go on the next time point? And guess what? I have a 1/10 chance of actually coming back into an A. So if I look at my alignment and I basically see, oh, great, in that species I have an A, and in that species I also have an A, I have to say, well, some of the time, that's going to be because I chose this 0.6 path multiplied by 0.6 path. And that's, of course, a much more likely path. But there's a non-negligible probability that, in fact, I chose the path of from A to C and from C back to A. Okay? So who's with me here on this whole concept of back mutations and how they need to be taken into account when correcting for what is the true number of actual substitutions that have occurred versus actual mutations that have occurred versus just the observed number of substitutions that it can actually account for. Okay, that's great. So we're at 100%, 0, 0, 0, 0. That's great. So this is basically the challenge that we need to face. And there are multiple models for estimating these back mutation events, but they're all based on the same idea of I have a Markov chain, and I can basically infer the probability distributions with which these nucleotides are changing, and from that infer the amount of time that has elapsed. So how do we evolve a nucleotide under a random model of mutation? Well, if we assume that, for example, you have 70% identity, or at any one time point, you can stay yourself with 70% probability, then that is a uniform model of substitution. That's basically saying that the A has 1/3 chance of going into each of the other nucleotides from a total of 30% chance of switching, but a 70% chance of staying. So basically with 70% probability, I stay who I am. And with 10% probability each, I go into each of the other nucleotides. So at time step 0, we basically start with nucleotide A. At time step 1, we can remain in A with probability 0.7, or we can change to C, G, or T with equal probability each. And where it gets really interesting is time step 2, where I can go back into A with probability either 0.7 times 0.7, which is the probability of staying, or back to A with 0.1 probability from each of the other ones. So that basically means that instead of having 0.49 total probability here, I actually have 0.52 probability of staying the same. And then here, instead of having 7 to the third, I have 0.412, and so on and so forth. Okay. So that is very important because over evolutionary time, that basically leads to me staying at the same nucleotide and converging not just at 0% identity, but at 25% identity. So this basically leads to this exponential drop-off of estimating the actual time that has elapsed. Okay, so let's explain and expand. So during an infinitesimally small time delta t, there's not enough time for two substitutions to happen on the same nucleotide. So we can estimate the probability of having a descendant nucleotide given an ancestor nucleotide and given a time elapsed delta t for any pair of nucleotides, and then let basically this probability of A given A given T, etc., be the matrix of substitutions. A reasonable assumption is that this is a multiplicative model, implying a stationary Markov process where the substitution matrix over time T plus T prime is simply the substitution matrix over time T times the substitution matrix over time T prime. Okay. Who's with me so far on this concept? So basically, we can expand the time and then sort of cut it into little pieces and then assume this multiplicative operation that allows us to now express longer time steps as products of shorter time steps. Okay, 88, 13, 0, 0, 0. So that basically means that the probability of going from y to x through time t plus t prime is nothing but the probability of going from y to z and then from z to x, summing over all intermediate time points, intermediate states z through this intermediate time point t prime prior to the t. So this way we can sum over all possible intermediate nucleotides. And what Jukes-Cantor model assumes is this constant rate of evolution. And therefore, for a short time epsilon, the substitution matrix over epsilon is 1 minus 3 times alpha epsilon, which is the rate at which I'm changing. And then each of them has only this epsilon timescale times this alpha, which is the rate of change. So this is a single parameter model. So Jukes-Cantor, even though it has two names, is a single parameter model. And that's basically encapsulating what we were talking about earlier with this uniform change from one nucleotide to the other. So for longer times, we can basically say that, wow, now there are back mutations. So basically the rate at which I stay the same versus the rate at which I'm changing to another nucleotide is going to be driven by this matrix, which I can now represent as collapsing, C, G, and T as this 1 minus 3 alpha of staying the same, 3 alpha of being another, and then 1 minus alpha of coming back to, 1 minus alpha of staying where I am, and another alpha of coming back. So using this, we can basically say that the probability of staying unchanged is 1/4, which is effectively the stationary distribution, times 1 plus 3, which is the 3 different options here, times e to the minus 4 alpha t, which is basically saying how well can I switch from each nucleotide to each other nucleotide. So basically, this is the probability of staying stationary along the diagonal. And then the probability of actually having a substitution is changing the A into any of these other letters. And what this results in, what this sort of geometric fall off results in, is that R of t starts out at 1 and then decreases all the way, starts out at 1/4 is basically where I will end up after infinite time. So basically 3 to the e to the infinity, that basically becomes 0. So these asymptotes to 1/4. And this other one also asymptotes to 1/4. But where are they changing? So when t is 0, this actually starts at 1. And when t is 0, this actually starts at 1/4. Okay. So basically this becomes 1 plus 3. And then this becomes 1 minus 1, 0. Okay. So basically the probability of staying the same is initially 1 and then dropping off all the way to 0.5. And the probability of changing is initially 0 and then eventually rising to 0.5. So that's one model. And we can basically now ask, given this model, and given that I have observed a certain amount of substitutions, then what is the actual distance that has elapsed? So basically, given a well-aligned portion of my sequences, xi and xj, I want to define the distance between the two sequences, which is going to be some kind of additive distance, as the fraction of sites where they differ. But that's not satisfactory because I'm not counting back mutations. But instead, a better model is to reverse that equation. So instead of saying, oh, great, over evolutionary time t, this is what my R of t looks like. What I want to know is what is the t given the number of substitutions that I have observed. So to do that, I just simply reverse that equation. So I'm taking the log instead of the exponent. And that's basically d of ij being minus 3/4 of the log of 1 minus 4 times the rate of the fraction of nucleotides that are identical divided by 3, which is basically this substitution rate that I have here. Okay. So when I apply now this model, this basically tells me, oh, in fact, that's directly from there. So that basically tells me that if I have an observed number of substitutions, I can infer the actual number of mutations that have occurred. And that's always an overestimate, because if I basically have only 0.1 divergence, I probably had another 10% more. If I'm at 0.2, I probably had another 30%. If I'm at 0.3, I probably have another 8%, and so on and so forth. All the way up to, if I'm at 0.7, I probably have had 2 substitutions. And then what happens after 0.7? What about 0.8? Well, 0.8 is unlikely because by chance, I can only diverge all the way to 0.75. So if I have less than 75% divergence, that means that I'm doing something wrong because I'm specifically excluding back mutations that would have otherwise occurred with the same frequency. So that's why this curve, basically when you get at 0.7, it starts increasing faster and faster. And when you get at 0.75, it basically asymptotes to infinity. So let's see who's with me so far on the back mutation estimation and on the correction for why the actual observed number, the actual number of occurred mutations is higher than the observed number of substitutions. Okay, so we're at 40, 60, 0, 0, 0. So that's the one parameter model that basically says that every nucleotide changes with the same probability alpha to each of the other nucleotides. The two parameter model basically says, well, transitions are much more likely than transversions. So I'm going to use a different rate for transitions and a different rate for transversions. So I'm going to have a higher rate S and a lower rate U. And now I'm going to have these C to T or A to G basically be expressed based on this smaller rate, and then these A to A, etc., based on the higher rate. And I think the alpha and the beta here are flipped. So we basically now have these A to G or C to T mutations based on this beta parameter, and then this everything else change based on these two parameters, basically alpha and beta, of sort of what is my chance of going to another nucleotide and then coming back. And then lastly, the remain, which is 1 minus the two transitions. So basically, we now have the ability to infer what is the amount of time that has elapsed given the observed substitutions that I'm observing and the number of mutations that I'm inferring. So basically, the amount of time can be thought of as, sort of a function more directly of the number of mutations rather than just the number of substitutions. So that's basically the Jukes-Cantor one substitution type model, so one parameter model. The Kimura two parameter model that basically has transitions and transversions. You could also have, with multiple substitution types, you can have one transition and two different types of transversions. Or you could have a fully symmetric model that is basically every nucleotide to every other nucleotide has, its own parameter. And that's assuming equal base frequency between the departure and the arrival or between two species that I'm comparing. Alternatively, I might actually have unequal base frequencies and therefore include models that, are, for example, a general time reversible model is a model that allows for these unequal frequencies. And, with one substitution type, but unequal frequencies, there's a model F81. With two substitution types, just like the transition transversion, but multiple different states for the frequency. There's other models and so on and so forth. So there's an entire hierarchy of models that exist for both nucleotides and peptides and codons. So Andrew asked, does this also hold for sequences diverging due to selective pressure?
I was asking about the observation that nucleotide frequencies trend towards 25% of the conserved.
I mean, of course not. So basically, if something is under purifying selection, it's basically going to stay there. And if it's something that's under positive selection, it's actually going to move, to zero. Does that make sense? So basically, if there's pressure to preserve a particular nucleotide, the blue curve is going to stay up there. But if there's pressure to diverge away from that nucleotide, then that blue curve is going to actually fall to zero.
I guess more what I was asking is, on an entire sequence of, let's say, 1,002 nucleotides, for example, that's being selected to have a new function, would you expect that the average substitution frequency to be greater than 75%? Does that ever happen, or will it in adopting a new function?
Okay. Yeah. I mean, I have to say that in terms of alignment, if you're searching for what is the orthologous sequence, and it's more than 75% diverged, you will never find it, except if you're using surrounding nucleotides to recognize it. So if you use synteny, as we talked about last time, and you basically sort of anchor your alignment, then of course you can catch something that's less than 75% divergent, sorry, more than 75% divergent, less than 25% conserved. But if it's, if you're just searching in the genome, you're much more likely to find something completely random that has 75% identity rather than something that has 80% divergence. Do you see what I'm getting at? So basically, by chance alone, if I search any sequence, I'm going to have 25% identity. Right?
That makes sense.
Yeah. Okay. And then transitions A to G and C to T, that's correct, Thomas. So A to G and C to T, these are transitions. Oh yeah, you're right. So yeah, no, that's what I mentioned earlier, that the colors are flipped here and the rates are flipped. So there's a mistake in the slide. Okay. So we basically talked about how we go from alignment to distances. How do we turn pairwise sequence alignments data into pairwise distances by basically computing a probabilistic model of divergence like Jukes Cantor or Kimura or this whole hierarchy of models that allows us to infer the actual number of mutations that likely occurred to yield the observed number of substitutions that we have between the pairs of sequences. Okay, so everybody with me on this first part? So let's see who's following. Lovely. Okay, so 70, 25, 5, 0, 0. Okay, so the next challenge is we now have a distance matrix. How do we turn that into a tree? And we're going to look at different tree building algorithms. First, we're going to define what are the different types of trees: ultrametric, additive, general, and then look at different algorithms: UPGMA, unweighted pair, geometric mean, or a group method with arithmetic mean, unweighted pair, group method with arithmetic mean, or neighbor joining, and what are their guarantees and limitations. And also we're going to talk about optimality methods, such as least squared error or minimum evolution, which actually require search that we have in section 4. So now how do we take the distance matrix between all of our pairs of alignments that we've now computed the distances of, and then map that distance matrix into a tree? So how do we infer a tree from a distance matrix? So basically one way to conceptualize what we're trying to do is that if I have 5 sequences, I can basically draw an arbitrary distance between any pair of those 5 sequences. So I can basically say I have dog and cat and human and mouse and rat, and I can just draw all of the pairwise distances and let them be whatever I want them to be. Okay? Everybody with me so far? So basically, this is just a fully unconstrained model. And that's basically the top part here of the matrix. That's basically saying, yeah, here's all the pairwise distances. Are we 100% together? Awesome. So 50, 40, 0, 0, 0. Now, the challenge, however, is that instead of saying, oh, I can go through any path, now I'm saying dog, cat, human, mouse, and rat can only travel to each other along corridors that are bifurcating. Okay, so previously I had a fully independent set of distances. Now I have a highly constrained set of distances that have to travel along the tree. And that's basically what I have here. So basically, this is one way of thinking of this spatial relationship between all of the different distances. So I can only travel from rat to mouse through this rat branch and this mouse branch. And if I want to travel to human, I better go through the same rat branch and then these common ancestors and so forth. So that basically imposes a constraint for each of those distances. This basically better be, to go from rat to cat, and I have to take R plus Y plus Z plus X plus C, okay? So we're basically constraining the distance to basically be only tree based. It's like, you have a nice grass field where a bunch of houses can travel just through the grass field, whichever way they want to go. Or you just draw a very specific set of connections through which everybody must travel to go between these houses. So, of course, these paths are much more constrained. And what we're going to be talking about is what type of distance matrix here at the top is in fact implying that the distances were sampled from a tree or not. Okay, so instead of having a random path between any pair of them, I can basically say, wow, it seems that the distances are indeed sampled from a set of from a tree. So we're going to be basically mapping the distances dij to a tree, and then searching for the set of parameters for d, c, x, h, m, r, y, and z, but also the topology that minimizes this total sum of distances between the total sum of differences, I should say, between the distances computed from the original alignment and the distances computed through the tree. Okay, so one way to do it would be to sort of find the set of parameters and the topology that minimize this total discrepancy between the observed distances d and then the tree-based distances m. Okay, so who's making me so far, and are there any questions? Okay, so 70, 30, 0, 0, 0. So we can now define conditions under which we know that the distances in a matrix are in fact sampled from a tree. And the easiest way to think about this is that if I, j, and k were sampled from a tree, if the pairwise distance were sampled from a tree, then there must be some assignment where the two of the distances are identical, namely I to k and j to k must be identical. And the third one must be smaller than the other ones. Because if it was larger, then I would have a different tree that, so basically, because I don't have a root, if these two were larger, then I would have built a different kind of tree topology. Okay, so what we basically have is that two of the distances are equal and the third is smaller as the condition for knowing that this was sampled from the leaves, that all paths from the leaves are equidistant to the root. That basically there's a root somewhere in here, and if the root happens in the root must happen in the longer branch because all of them are actually equidistant. So that's why the longer branch must be the one where the root is, and that's what this is telling us. Okay, so this is corresponding to a rooted tree with uniform rates of evolution. Okay, so is everybody following on this whole ultrametric tree? So an ultrametric tree is a tree that is resulting from a chronogram where the distances of all extant lineages to the root is actually the same. So 44, 33, 22, 0, 0. Okay, so that basically implies always that the root is at the midpoint, and if A is greater than B, then the root flips side, and that's why the A and B will swap. There's a more general condition which does not require that the distance from the root to the tree to the leaves is the same for all species, for all lineages, and that's that the distances satisfy this 4-point condition, that any quartet can be labeled ijkl such that dij plus dkl is smaller than dik plus djl, and that's the same as dil plus djk. And the reason for that is simply these, if you map the ABCD, these distances simply add up. And the result of that is that all pairwise distances can be obtained by traversing a tree. Why are these conditions interesting? Because you don't have to worry about knowing the actual values. All you need to do is test whether they satisfy this condition, and then you're guaranteed that there exists a tree that satisfies that, but you don't have to actually construct a tree to figure it out. So that's why knowing whether a distance is ultrametric or additive is actually a very helpful construct. Okay. And then, of course, here the root can be anywhere, but because we don't actually have to have the same distance traveled from the root to any of the leaves. And of course, the third type of distance is just general distance matrices. So in practice, a distance matrix is neither ultrametric nor additive. One of the reasons is noise, because we're measuring distances that are just not exact. Another is the evolutionary model that we're using to compute these distances, whether it's through Cantor or cumulative parameter model, they're just simply not exact. And also there's fluctuations. The regions that are used to measure the distances might not be representative of the species tree, or there might be gene replacement and gene conversion or lateral transfer, or simply varying rates of mutation, which can lead to discrepancies. But in the general case, the tree building algorithms must handle noisy distance matrices, and you can obtain such a tree either by enumeration or scoring of all trees, which is too expensive, or by neighbor joining, which typically gives a good tree, or by UPGMA, which typically gives a poor tree, unless the distances are ultrametric. So now let's dive into these algorithms. So now that we have conditions for these distance matrices that basically tell us whether they are sampled from a tree or not, then let's look for algorithms that reconstruct those trees. So UPGMA, or hierarchical clustering, is basically this unweighted pair group method with arithmetic mean. And the procedure is identical to what we had seen in the hierarchical clustering method. Namely, we're finding two clusters that have minimal distance between them, and then we're taking the union of these two clusters and defining a node, connecting them and placing it at the height, which is halfway between these points. And then we're deleting the initial clusters, and then we're now computing the distance between that new 2-3 cluster and the remaining clusters in our tree. And every time two clusters, when only two clusters remain, then you place the root halfway between them. So here's an example. We basically have these human, mouse, rat, dog, cat distances, and we have this full distance matrix that basically says 12, 12, 12, 8, 8, 8, etc. And then we can basically start merging together pairs of distances, pairs of species that are the closest to each other. And here, rat and mouse are the closest, so I can merge those first, and then put the point halfway between them, and then recalculate the distances. And then the next one after that is cat and dog that are only 4 apart. So I can sort of merge those two, and then again put the halfway point between them at 2, 2. And then the next one after that is 7 between dog and cat. So then I can merge, dog and cat, and then mouse and rat, and sort of put that at 7. So basically that's 2 plus 2, 7. So anyways, halfway between those. And then the last one is human, where I can basically sort of place the root wherever human is. So basically, that's great. The distances were not adding up, because it's actually that cat and dog are getting merged together with human, and that's what gives me the 7 there. So basically, I have 2 and 2, and then 1 and 5, and so on and so forth. Okay, so this is just a simple procedure for how do we merge these pairs of distances together. So first we merge mouse and rat, and then we recalculate the distances. Then we merge dog and cat, and we recalculate the distances, and eventually we get to the tree. Okay. So this algorithm, UPGMA, unweighted pair group method with arithmetic mean, is guaranteed to build the correct tree if the distance is indeed ultrametric. And the reason for that is that the tree topology is unique, given that the tree is binary, and UPGMA constructs a tree obeying the pairwise distances. Therefore, because it's unique, it must be the correct one, and because it matches. But UPGMA has a problem. It basically assumes that there's a molecular clock that basically from the common ancestor, all of the species have undergone the same number of mutations as if they all had the same mutation rate. So it implies that the time or the speed of divergence is actually constant for all species. But of course, some species, such as, for example, mouse and rat, evolve much faster. And UPGMA will basically, make incorrect trees for those. So if, for example, my tree is this one, where basically species 1 in this subtree evolved super fast, and species 4 in this subtree evolved super fast, then when I calculate my UPGMA tree, what I'm going to be doing is finding the shortest distance and then merging those together. So 2 to 3 are in fact the shortest distance. So that basically means that I will group 2 and 3 together and then recalculate their distances to 4 and then their distances to 1. So even though this was the correct tree, UPGMA will try to make the tree that assumes the most uniform rate of evolution from a common root. And of course, that's obviously the incorrect tree compared to what it should have been. So how do we combat that? Well, one way to combat that is to say, well, maybe I could look for the fact that 2 and 3 have short branches because they're in fact closer to all the other species. And maybe 4 and 1 have longer branches because they're further from all the other species. So instead of worrying only about pairwise distances for any one of them, what I'm going to do is calculate an overall distance between any one node and all of the other nodes. And that's effectively what neighbor joining does. Neighbor joining is guaranteed to produce the correct tree if the distance is additive, and it may still produce a good tree even when the distance is not additive. And the key idea is that we're going to be computing a new distance matrix where I'm going to have some kind of promiscuity score for every one of these leaves. So instead of taking the original Dij distance matrix, I'm going to be taking Dij and subtracting from it a promiscuity metric that basically tells me how connected, what is the total distance between every node and every other, between every node of interest and every other node in the tree. Okay, so we're basically computing an overall distance that basically tells me, across all other pairs of nodes, so basically from Di to any other node, what is that overall sum distance normalized by the number of leaves, so the average distance between D and all other nodes. Okay. Is everybody with me here about the difference between neighbor joining and UPGMA? So basically neighbor joining has this normalization factor that takes this global promiscuity into account. So it has, this R that basically says, hey, is this really proximal to all of the other ones? And if so, then I will scale the distance accordingly. So we're at 60, 40, 0, 0, 0. So the claim is that the above magic trick ensures that Dij is minimal if and only if I and J are neighbors. And the proof is actually beyond the scope of this lecture, but you can find it in the Durbin book in page 189. So the neighbor joining algorithm is therefore you initialize T to be the set of leaf nodes, one per sequence, and then the set of leaves is T. And then you pick IJ such that now the capital Dij is minimal. And then you define a new node and you set DKM to be 1/2 times this set of distances for all M. And then you add this new node K to T with edges of length this, which is including that adjusted distance. And then you remove that node from these pairs of nodes from the original set. And then you now add the new combined node. And then you iterate until there's only 2 nodes, and then the edge between them is of the appropriate length. So this is exactly the same algorithm as UPGMA, only we've changed the way that we're computing the distances to now include this additional correction factor. So what have we seen so far? We basically looked at these 2 ways of constructing trees using clustering from distances. We looked at UPGMA and neighbor joining. With distance-based algorithms, we can also aim to directly minimize the discrepancy between the original distance matrix and the tree-based distance matrix. And that's, for example, what least squares does. It basically says, what I'm going to do is find a topology that simply minimizes the discrepancy between the pairwise distance, which is unconstrained, and the tree-based distance, which is constrained to walk through the tree. So that's what least squares, for example, does. And then minimum evolution is very similar. It just tries to minimize the total amount of evolution that has occurred given the tree topology that I have. Okay. So this is all based on distances, and the clustering algorithm simply has some heuristic, some recipe that you follow, whereas this optimality criterion of least squares is specifically searching for a tree that fits a criterion, whereas these guys just follow a recipe and hope that most of the time it'll lead to good trees. So what we basically talked about is how do we now take this distance matrix that I built from my alignments and then take these distances and turn them into trees. And we saw two different tree building algorithms. We saw UPGMA and neighbor joining, and also the guarantees and limitations relative to whether a tree is an ultrametric tree, which basically guarantees that UPGMA will be correct, or an additive tree, which guarantees that neighbor joining will be correct, or just any general tree for which neighbor joining will generally produce a good starting point. And then we talked about this optimality criterion for building trees based on least squared error or minimum evolution, which requires search, as we're going to see in section 4. All right, so who's with me here so far on sections 1 and 2? So we basically saw how we can turn alignments to distances and how we can turn distances into trees. Okay, so we're at 60, 40, 0, 0, 0. Now let's turn to how do we directly infer trees from alignments, and namely, how do we score an alignment given the tree? So we're now going from alignments directly to trees without passing through this intermediate of distances. So we're going to look at a parsimony method, which is set-based, a parsimony method using dynamic programming, and a maximum likelihood method for evaluating the goodness of a tree given an alignment. So again, instead of sort of going first to distances and then to trees, we're now going from alignment directly to phylogenies by coupling them, as we were going to see in section 4, to tree proposal and sequencing. So character-based phylogenetic inference is really about tree scoring, not tree finding. We have to couple them with a tree proposal and update, and then you have an algorithm, which is part 4 of the lecture. There's two approaches, and they both use the same architecture. The goal is to minimize the events using parsimony, such as union intersection, or to minimize the cost according to a maximum likelihood or a maximum a posteriori model. So now let's look at the simple version of parsimony scoring. So parsimony scoring basically says, given a tree and given an alignment column of that tree, for example, ABAB, we're going to label the internal nodes to minimize the number of required substitutions, okay? And this algorithm works as follows. So initially we have cost 0 because we have 0 events in the whole tree, and then, some k. And now we're going through every single node. So if k is a leaf, then we basically set this to be the union, the state of this to be the union of all characters at that leaf, okay? So basically, for every leaf, we basically have a set, which is only containing one character. And now we're just going to do this union intersection decision. So if k is not a leaf, we now have this. Then let ij be the two daughter nodes, and let our k be the intersection of the two sets. If that intersection is non-empty, namely if there is a common character, then I'm going to choose that common character. But if there is no common character, then I'm just going to set it to be the union of the two. Okay. So walking up the tree, I basically say, well, I have an A here and a B there. So that basically means that this node is either an A or a B. And in either case, the cost is going to be 1, because if it's a B, I'm going to have to pay a cost of 1 substitution towards the A. Or if it's an A here, I'm going to have to pay a cost of 1 substitution towards B. And then I go up the tree. And then I see that, wow, here I know that it's either A or B, and they both give me cost of 1. And here it's clearly an A. So I'm going to choose the setting of that variable that minimizes the cost, and that is simply A. And I don't have to pay anything else. So that cost is not incremented. Okay. So if the intersection is empty, then I pay an additional cost. But if the intersection is not empty, then I don't pay an additional cost. And then I basically go further up and I say, well, on one side for this node, what is the ancestral value of that one? If I have an A here and a B there, that must be either A or B, and then I pay another cost of 1. Okay. So who's with me so far on this union intersection algorithm for parsimony? Okay, lovely. So we are at 70%, 30%, 0, 0, 0. So this is nice and lovely, and it basically tells us the cost of a particular column of the alignment given the topology. So notice that all we're doing now is scoring the topology. We're not searching for topologies. The way that we're going to search for topologies is going to be in section 4. But right now, we are scoring a topology, and we're not scoring a topology across the entire sequence. We're scoring a topology 1 column at a time. So to turn this into a full algorithm, what we need to do is just score that topology for every column independently, and then put an outer loop where you search over all possible topologies, and then you choose the topology that minimizes the total cost of generating this. Okay. So the problem, of course, with parsimony using this union intersection is that not all possible values are accessible. So basically, this traceback could say, okay, here's A, B, A, B. I must have, so for example, I could have A or B here, but I must have A there, and then I could have A or B there, and so on and so forth. But there's another assignment. Basically, if I have this kind of union intersection result, like we saw in the previous slide, I could basically resolve it by saying, well, either the ancestor is B, and then it changed into an A, and it stayed an A, and then it changed to a B here. So there was 1 substitution there, and then 1 substitution here. Here I went from B to A, and here I went from A to B. There's another possibility, which is I can label the ancestral node A, and then I have a score of B here, a cost of 1 to change into a B, and then no cost here, and then another cost of 1 to change into a B here. But that basically does not capture this particular solution, which is still optimal, but never found by this traceback, because I basically could have, because of the way that parsimony works in this union intersection, it basically makes a greedy choice. And it basically says, I will not consider anything else over here rather than just the simple intersection of A, B, and A. But if I had any knowledge that outside there's another B, then a B here would have been equally plausible, equally, same reward, same cost. Okay. So that BBB is still optimal, but it's not found by the tracebacks. It's not possible. But there's another way of doing this parsimony scoring, which is, why bother with only intersection and union if I could instead maintain a score for every possible setting of these ancestral nodes? So I could basically say, well, given that I have A and B here, if I have A here, my score will be 1, and if I have here, my score will be 1. And then over here, instead of just saying, oh, it must be A, I can just say, well, if I have an A, it has a score of 1. And if I have a B, it has a score of 2. So now I can maintain that score of 2 and continue propagating it up at the same cost. And then now if I have an AB, I can just say, well, if it was an A, I would have a score of 1. If I have a B, I have a score of 2. But then the B suddenly has only a score of 1 here, and the A has a score of 2. So now A and B are equally plausible and equally costly at the root. Okay. So let's now go through this with a full dynamic programming matrix that I can, compute from the bottom to the top. So I have my initial leaves for mouse, rat, human, and dog. And I have a score of 1 if I set them to anything except the value that they have there. So their value of mouse is A. So everything has a score of 1, a penalty of 1, except for A that has a penalty of 0. Okay, so now I'm asking how many events do I need to explain the tree in any possible way? For rat, same thing. Rat has a character of G. So everything has a score of 1 except G. And so on and so forth for A, for human, and for dog. And now I can basically say, what are the possible scores for this internal value B1? The possible scores are either 1 if I change into an A, or 2 if I, set it to be a C, or 1 if I set it to be a G, or 2 if I set it to be a T. Namely, if I set it to anything but these two values, then I'm going to have to pay an extra cost for changing here and here. Whereas if I set it to one of those two values, then I pay no extra cost. Is everybody with me here on this column? So basically what I'm doing is I'm using these penalties to basically compute what would be the minimum score that I can have given the children of every one of those nodes and given the cost that I would be paying to change into each of those. So we're at 67, 33, 0, 0, 0. So the same thing I can do, just continuing to walk up the tree. So basically, I now have propagated the scores for every setting of that B1 variable to A, C, G, or T. And then I can just say, well, given that A has this cost structure, what is the cost structure for B2? And that's a cost of 1 if it's an A, a cost of 2 if it's a G, and then a cost of 3 for any other setting. And then incorporating the dog as well, I can basically say, well, the dog would have to pay a penalty unless it's a G. And therefore, the additional score all the way to the top is simply, the sum of those guys. And therefore, I can have two different settings for B3. And that's either an A with score of 2 or a G with score of 2. And anything else has higher scores, so I'm not interested. So every cell represents the minimum cost of the subtree rooted at N if the label at N is C. And I update the table by walking up the tree from the leaves to the root, remembering the maximum choices that I make. And then I trace back from the root to the leaves to construct the minimum cost assignment. So this is basically dynamic programming going up the tree. Who's with me so far on this whole algorithm? Very cool. Okay, so we're at 70, 20, 10, 0, 0. So we are now ready. We have all of the methodology ready to start tackling the machine learning view of parsimony scoring, of alignment scoring. So instead of working with these discrete steps and these scores, we're now going to build a full probabilistic model that includes back mutations and lengths of trees and include this peeling algorithm from Felsenstein. So this is basically scoring using maximum likelihood and maximum a posteriori. So the input is going to be your sequence alignment, and your output is going to be the tree with maximum likelihood or maximum posterior probability. And of course, we're going to have a search of a heuristic searching for the maximum likelihood tree. So what we're going to be focusing on this section, before we get to 4, is how do we score a tree using maximum likelihood. So this is the maximum likelihood model. It basically says, I have a set of branch lengths and a topology, which are going to be maximizing the search over all branch lengths and all topologies, such that I maximize the probability of observing my data of the sequence alignment, given the branch lengths and the topology. The maximum a posteriori basically says instead what I want to maximize is the posterior probability of B and T given that I have observed the data. This is the generative probability. That's the forward-looking arrow that we were mentioning in the beginning of the class. And then this is the backward-looking arrow. And how do we get this backward-looking arrow? By applying Bayes' rule and basically saying that B of BT, probability of BT, the branch length and the topology given the data, is nothing more than the probability of the data given the branch length and the topology times the prior of the branch length and the topology, and they should say prior, not likelihood. Okay, so that's the prior, that's the likelihood, that's the prior, and that's the posterior probability. Okay, so, and the reason why I can ignore the divided by B of D is because this probability of all of data is the same regardless of what hypothesis I choose. So basically, as I'm searching for the maximum, it doesn't depend on D, so I can just get rid of that. Okay. So basically, this using Bayes' rule is that, and that I can just ignore that. So this is nothing more than that. So the probability of the data given the branch length and topology is the likelihood of the data given the model. And we can use the sequence evolutionary model, Jukes-Cantor, cumulative parameter, or HKY, that basically give us directly the probability of observing a set of distances given the branch lengths and the topologies. The probability of the prior probability of the branch lengths and the topology is the prior, and we can use a generative model to construct the probability of that prior. So we could have, for example, a birth-death process that we could model for how likely is the topology that I'm observing and the branch length that I'm observing in absence of any data. And that's just a prior. And that prior can be given by an evolutionary process that we're running, a simulation process that is basically saying, you know, how often do I gain a branch, how often do I diverge, how often do I mutate, and so on and so forth. So this is basically just giving us a prior probability on a set of trees and a set of branch lengths. Okay, so because we've been here on the difference between this maximum likelihood model that is looking at the data given the branch lengths and topology versus this maximum a posteriori, which is basically looking at the data given via T times this prior of these topologies. Up. All right. So we're at 70, 30, 0, 0, 0. So what we're going to be doing now is we're going to compute this P of D given BT, and that's the same whether I'm using maximum posteriori or maximum likelihood, because the only difference is this prior here. So let's now compute the probability of the data given the branch length and the topology. So this is a generative model. This is basically saying how likely am I to generate these data given the branch lengths and the topology, and we're going to be computing that recursively using dynamic programming. Okay, so this is basically Felsenstein's peeling algorithm for how do we compute this probability of the data given the branch length and the topology. So suppose that I have some sequence AAA CTG in the ancestor. My question is, how likely am I to generate different types of sequences from that ancestor? So that's basically the total probability of observing a vector of all of the internal nodes given the overall topology and the lengths of time. And that's basically making the assumption that all of them jointly, this joint total probability, is simply x1 given all of them times p of x2 given all of the rest of them, and then x3 given all of the rest of them, and so on and so forth. And x1 given all the rest of them is nothing but x1 given the parent of x1, and x2 given all of them is nothing but x2 given only the immediate parent of 2, and x3 is again only 3 given the parent of 3. Why is that important? Because we're basically assuming this branch independence. We're basically assuming that every branch is independent, conditioned on the parent. So that allows us to now simply take this super complex object and then represent it as each of them based on its parent, which is nothing more than the probability of that root, which is the prior probability of having that particular sequence at the root, times the product of each of the children given the parent and the time t that has elapsed from that parent. Is everybody with me on sort of this simplification by assuming this branch independence, where we basically can simply express every leaf or every internal node as a function of its parent and the time elapsed? Awesome. So we're at 70, 30, 0, 0, 0. So let's assume basically that every site evolves independently, so that every column of the alignment is in isolation. We assume branch independence, condition on the parent, and therefore expand the total joint probability into the product of P of xi given the parent, given the time elapsed for each of the branches. And then only the root remains, which is simply the root prior, and that's simply the background nucleotide frequency. So that's nothing but what is the probability of having an A, C, G, or T in every column of the alignment at the parent, all the way to the root. And then we actually know how to compute the probability of a child given its parent and the time t, right? That was the first part of the lecture. This is exactly the sequence model that we built for Jukes-Cantor or the cumulative parameter model, the HKY model, and that whole hierarchy. So we can actually calculate for any given assignment of internal nodes that child from parent probability given the time elapsed. And as the internal node values are not known, what we're going to be doing is marginalizing. So we're going to be summing over all possible values of all internal and root nodes. And then all of these are going to be representing the n-1 internal nodes. So we're going to be iterating over all possible values to basically say with what probability have I generated the observed sequences. So instead of making an assumption of the maximum likelihood estimate of the ancestral node, we're going to be iterating over all values. So what this algorithm is doing is effectively giving us the total probability with which the observed data of every column of the alignment can be generated from the evolutionary process that's moving along these topology lengths, this topology and these branch lengths. Okay, so this is basically scoring a particular topology, iterating over all ancestral values and evaluating the total probability with which I can generate this. Okay. So then we've already talked about this Jukes-Cantor model at the beginning, where we basically have these, asymptotic convergence to 0.25. We've talked about how to separate each column of the alignment into a product of individual bases, assuming site independence between those bases. And then how we can actually compute the sequence evolution over the entire tree by assuming branch independence, by assuming a prior on the root sequence, and then, for example, 1/4 probability for every sequence length, M for every nucleotide. And then we can use the product and the prior to define sequence evolution over the entire tree. And then lastly, we integrate by marginalizing over the hidden ancestral sequences. So notice all of the sequences are needed, both internal nodes and leaves, but only the leaves are given. So therefore, we need to marginalize to sum to integrate over all of the unknowns, which are the internal sequences. And yes, this looks expensive because we are supposedly iterating over all of them, but we can actually use dynamic programming in the same way that we saw for the parsimony model, where instead of searching over every single possibility one at a time, we can basically propagate the scores and then remember the maximum choices and then do a trace back when we get to the root for what is the maximum probability. So we're going to be basically summing over all possible values of the internal nodes. We're going to be summing over x5 or x6 and x7, and then iterating over that. And then this is basically this dynamic programming algorithm. So basically this length of I and J and A will be the dynamic programming table. Every entry will contain the probability of seeing the leaf data node below node I, given that node I has a base A at site J. Okay, so basically this is between every parent and its child, given the base position. So it's basically L, the number of leaves, n, 2n minus 1, which is basically the total number of nodes, times the potential characters at each of those. And then the leaves of the table are initialized based on the observed sequences, and then the entries are populated in post-order transversal, which basically means going up the tree. And the runtime is nothing but 2n, which is the total number of leaves, times k squared, which is all of the possible assignments for every single pairwise change that I can make to go from each character to every other character. So k is the total number of characters. And basically this probability is given by, these leaves that are either 1 or 0 if they're observed, and, that probability otherwise. So we can now use dynamic programming to compute the maximum of all of these functions recursively up the tree. So we're starting with the leaves and we're going up the tree, and every single time the two children of every node are basically sending information, and that's where the k square comes in, because I'm basically asking what is the probability of going from character A to character B and from character A to character C, and then I'm multiplying over these total probabilities and then summing over all 4 nucleotides and all 4 nucleotides here. And that's what basically leads to that total probability. I'm taking the maximum over all of that. So if we know the branch lengths T left and T right, we have already the likelihood tables of the left and the right subtrees for every possible ending character at B and C. So all we need to do is fill in the likelihood table for each remaining character A. So basically we initialize using the extant sequences that are observed, and then we're moving up the tree from the leaves to the root. And the characters at the leaves are not already known. Are already known. The likelihoods are 1 or 0, indicating the known character. So what we do is that we fill in the internal node likelihood vectors iteratively, and when we reach the root, we just multiply by the base frequencies and then maximize over the topologies and the lengths. And that's where we basically do gradient descent or searching or Newton's method or anything else in order to basically search over this. So the advantages of these maximum likelihood and maximum a posteriori methods is that they are inherently statistical and evolutionary model based. They're usually the most consistent methods available, and they're used both for character and for rate analysis, and they can be used to infer the sequences of the extinct ancestors by integrating over them or by finding the maximum likelihood state for those, or the maximum a posteriori state for those. And it accounts for both branch length effects in unbalanced trees, and also the nucleotide or amino acid sequences work equally well, and any other kind of data. The disadvantage is that it's not as intuitive as parsimony. So you might choose more events if they're more likely in the probabilistic model, and they're computationally very intense. So this limits the number of taxa and the sequence length that you can use. And just like parsimony, they can also be tricked by high levels of homoplasy, as basically if you have convergent evolution. And any violation of the model assumption can also lead to incorrect trees. So what have we seen so far? We've basically seen how we go from alignments to distances, how we go from distances to trees, and how we go from alignments to trees. All that's missing is this tree exploration tool, which is basically this tree proposal and scoring iteration loop. So this is the outer loop of the whole algorithm. So basically the heuristic tree search for a character-based reconstruction is initialized by some tree. And given how well neighbor joining performs, we usually initialize with a neighbor joining tree. And then we basically do a Markov chain Monte Carlo search. We're computing the probability of this tree, and then we're computing either nearest neighbor interchange or subtree pruning and regrafting. And nearest neighbor interchange, basically taking any quadruple and then swapping either A with C or A with D. And subtree pruning and regrafting is basically taking any subtree and then regrafting it somewhere else. Okay, for example, C, instead of being on this branch, can now go on that branch. So we can basically modify the tree using these operations and then calculate the score of the modified tree and then choose to accept or reject based on the difference in likelihood between the modified tree and the original tree. So we propose many trees using rearrangements. We score each of them, and the score can be parsimony or likelihood or posterior probability. And the algorithms can differ on how they score, how the score is used to guide the tree search. So using Markov chain Monte Carlo, you can explore your state space in a way that makes it sample from the posterior after skipping some burden in and only taking 1 out of 100 to make sure that the samples are independent of each other. So we've talked already about this in Gibbs sampling and other approaches. So if you convert any proposal rule that is a connected one into one that visits the states according to their posterior, the rule is if it's better, always take it. And if it's worse, you basically compute the likelihood ratio between the probability of the proposal and the probability of the current. And with some probability, the next becomes the proposal, even if it's worse. But that probability is dependent on that likelihood ratio. The more like the better one it is, the more likely you are to pick it, even if it's not the best one. And the ratio is because we don't need to compute the evidence, P of D, and that's why we only focus on the ratio. So while any connected proposal rule is okay, in practice, the proposal strategy is an art, and you need both good mixing, where big steps are good, but you also don't want to fall off the cliffs, so small steps are good. So you want to ensure a low rejection rate so that your step size is not so big that you typically fall off the cliffs. And a good rule of thumb is try it, see if the low rejection rate is low, and then it's good. So you can basically compute that ratio, and then, according to that particular ratio, you can make the specific decision of keeping it or not. The last concept that I want you to grasp is bootstraps, which is basically asking if I resample my alignments randomly by choosing columns with replacement, and I create many alignments of equal size, then I build a phylogenetic tree for each of the samples, and I repeat this many, many times, like thousands of times. I can then output a summary tree, which is the tree that's constructed the most frequently, or a consensus tree, even if it's not the most frequent, or some other option. And then the key part of bootstraps is I report the observation frequency of each branch. So every branch is a binary split between this and all of the other ones. And that branch basically can tell you, well, this branch was recovered 100% of the time. So I'm really confident about this branch. But this branch here was only recovered 49% of the time. So I'm not going to put too much emphasis on this one. It's probably going to be, incorrect. So that's where this bootstrapping comes in. So you can basically say, well, with 1,000 samples, this tree was always keeping those this way, but only 30% of the time keeping those that way. Okay. So who's with me on the tree proposal in search and also on this bootstrapping methodology? Okay, very cool. So we're at 60, 30, 10, 0, 0. And then we basically covered the whole lecture. We now went from distance-based methods that first compute a pairwise distance matrix from the sequences and then build trees from that, or these character-based methods that basically score what is the total goodness of a tree based on the evolutionary processes that it would imply at every column of my alignment, given this iteration over all possible ancestral states, and then how we could couple that with a tree proposal and scoring algorithm to search all that. So there's a lot of additional references that I encourage you to look at. This is UPGMA, least squared errors, maximum parsimony, maximum likelihood, neighbor joining, maximum a posteriori, and then some of the latest work on gene trees and species trees, and so on and so forth. Okay, so that's the lecture for today. We talked about the basics of phylogeny and then different ways of building them, from alignments to distances and distances to trees, or from alignments to trees, and this whole tree exploration and Markov chain search. All right, last question is, who feels that they've learned stuff today? Let's see. So we're at 44, 56, 0, 0, 0. Awesome. Well, thanks everyone. And then see you next time. Bye-bye.